Thank you very much, Sharon. Thank you very much, Runsit. Really, it's such an honor to be here. I feel like I'm a little bit of a fraud because I am actually a hemoncologist by training. I'm a stem cell transplanter, and my interest is in immunotherapy. But like so many of you here, I've also been personally really affected by ovarian cancer in that I've had two of my closest friends develop ovarian cancer. Actually, my best friend in the world, 6 weeks ago, was diagnosed with ovarian cancer, and really my lab's focus now is very much on trying to develop immunotherapy for this disease. So just let's take a little bit of a step back, and I want to tell you about our immune system and how amazing our immune system is. Um, so our immune system, our white blood cells, they have evolved to protect us from infections, of course. They protect us against viruses, against bacteria. But our immune system is also very powerful in that it continuously patrols our body to look for abnormal cells, for transformed cells such as cancer cells. So actually our immune system protects us both against external enemies, infections, and internal enemies, such as cancer cells. The problem is that cancer cells find ways to camouflage themselves, and they find ways to hide from the immune system. So the field of immunotherapy, basically, as the name suggests, it is the field that focuses on trying to harness the power of the immune system to fight cancer. So how do we do that? We can do that with drugs, or we could do that with cell therapy, and that's what I'm going to focus on, and that's the focus of my research. So cell therapies that are also often referred to as living drugs, and you will see in a minute why they're called living drugs. What they do is to take the patient's immune cells. And for the purpose of this presentation, I would like you to remember the name of two immune subsets, T cells and natural killer cells. And again, I'm going to come back to that. So what we do in cell therapy is we take the patient's, or as I will show you, a healthy donor's immune cells out of the body, and in a very specialized lab, these cells become trained to recognize those cancer cells that are camouflaging themselves and they are hiding. So basically, just to go a little bit deeper, it's like you take these immune cells out, and by genetically engineering them, you give these immune cells a GPS system. And this GPS system is called the chimeric antigen receptor, or a CAR for short. And through this CAR, now this immune cell is capable of recognizing the cancer cells that have camouflaged themselves. And what we do is we grow these immune cells, increase their numbers numerically, and then these are given just like a blood transfusion to a patient. Inside of the patient's body, these CAR-engineered immune cells grow, they seek out the tumor cells, they fight the tumor cells. And so there are actually currently 7 FDA-approved CAR T-cell products for blood cancers. So patients with B-cell lymphomas, B-cell leukemia, and multiple myeloma. And recently, these cell therapies are also being used more commonly for the treatment of solid tumors, and solid tumors also include cancers such as ovarian cancer, breast cancer, etc. And so the field is moving from using these cells for hematologic malignancies, blood cancers, to solid tumors. However, as you can see from this cartoon, despite the fact that these FDA-approved products do exist, there are limitations to their use. Even in the setting of blood cancers, only around 20% of patients who could potentially benefit from these cell therapies are gaining access. And the reason for that is manifold. One is, as you can see in this cartoon, The T cells are made from the patient for the patient. So this is a single patient product. And with this comes the complexity of manufacturing a drug for a patient that can usually take a number of weeks. And also the cost. I mean, the cost of one dose of these cell therapies is in the region of $500,000. Despite the fact that these cells are engineered just to seek out the cancer cells inside of the patient's body, they produce inflammation. Patients can become sick. They can develop high fevers. They can require high levels of oxygen to help with their breathing. They can drop their blood pressures. They can develop neurotoxicities. Somewhere in the region of a third of patients will need treatment on the intensive care unit. So these are therapies that can only be delivered in very specialized centers, such as MD Anderson, for instance. And of course, not all patients have access to big cancer centers where these therapies can be given. So because of all of these, only a small fraction of patients benefit from or gain access to these therapies. So of course, then the question is, if these therapies work, and we've demonstrated that at least they work very well for a subset of cancer types, how can we make them more commonly available, and how can we increase access to these therapies to more patients? Well, ideally, you'd want to have a cell therapy that is effective. You'd want the cell therapy to be safe. And we would want these therapies to be off the shelf and scalable, just like drugs, just like antibodies or chemotherapy that you heard about. We would want to have these cells ideally manufactured at scale so that when a patient comes, we can just grab it off the shelf, infuse it to the patient. And we want to have access to these therapies that's equitable. This shouldn't be determined by where you live and what's your nearest hospital. And we want these therapies to be logistically simple to administer, ideally in the outpatient setting, rather than having to bring patients in. So a number of years ago at MD Anderson, we asked this question: what if instead of using the patient's own immune cells to manufacture the product, we take immune cells from a healthy donor? And then we manufacture these cells in the lab and we engineer them with the CAR. So we introduce the GPS system, and then we freeze these cells at scale, such that when a patient comes, then the cells are available for off-the-shelf use. Of course, this would be ideal. The problem is you cannot do that with T cells, because you cannot safely transfer T cells from one donor, from one person, and give it to another person. If you were to do that, it would cause a very toxic syndrome called graft versus host disease that can actually be fatal. But remember how I mentioned our immune system comprises of different cell types. And one of these cell types is called natural killer cells. And natural killer cells, which for short we call NK cells, as the name suggests, they are actually very good at recognizing and killing virally infected cells and tumor cells. But the beauty of natural killer cells is that you can safely give natural killer cell from one person to another, and it will not cause graft versus host disease. It will actually not cause any toxicities. So then we ask this question, well, what if we take natural killer cells and we engineer them and introduce a chimeric antigen receptor? So we give them the GPS system, and then we manufacture them at scale, and then we make it available for patients. Then the next question we had to ask was, then who should be that donor? You need to know how to pick your donor if you're going to make a biobank of 100 plus doses. I mean, do you want to go and get someone off the street? Do you want to get the patient's sibling or spouse or parent, etc. And so as it happens at MD Anderson, we have an umbilical cord blood bank. So umbilical cord blood is a byproduct of birth, is usually discarded after a lady gives birth to her baby. And around 2 decades ago, my good friend Dr. Spahn established a cord blood bank at MD Anderson in partnership with the women's hospitals in Houston, but also across the United States. As you can see here, these are our partners. And we've now collected and stored over 40,000 cord blood units. And this bank was initially really started for cord blood transplantation. Cord blood is a very rich source of stem cells. As it happens, though, cord blood is also a very rich source of healthy, happy baby natural killer cells that are very good at killing cancer cells. So we developed a strategy where we showed that from one umbilical cord blood unit, in a 2-week manufacturing process, we can manufacture more than 100 doses of these CAR-engineered natural killer cells. When we calculated the cost of one dose of this therapy for us, costs around $600 to $1,000, so significantly less than the $500,000 of CAR T cells, which automatically would make this much more accessible and much more available. Of course, then the question was, are these CAR NK cells capable of recognizing and killing tumor cells? So this is a video showing one natural killer cell. This is one CAR NK cell in green. These are tumor cells in red. And when the tumor cells die, they will turn this magenta color. But just to show you how this NK cell comes and kisses this one tumor, kisses the second tumor, transfers the toxin into the tumor cells and kills them. So these natural killer cells are actually serial killers. So it's great. We show in the lab that they're capable of recognizing and killing tumor cells. Well, of course, the question was, would they work in patients? So we started a clinical trial. And the first clinical trial that we did was in the setting of B-cell malignancies. Remember, I mentioned that CAR T cells are FDA approved for B-cell malignancies. And these CAR T cells recognize a molecule that B-cell cancers have on their surface, like a beacon. And the name of this molecule is CD19. So the first thing we did was to make our CAR NK cells to recognize CD19, because we figured if it doesn't work against B-cell malignancies, well, it's not going to work against anything else. You know, that was like our control. And so we started this trial back in 2016 at MD Anderson. And this is what we call a PET scan. A PET scan is a total body scan, as you can see here, where we inject a radioactive dye that is taken up by cells that use up a lot of energy. So cancer cells, obviously, because they're growing, they use up a lot of energy and a lot of glucose, etc. Our brain always uses up a lot of energy. So this is normal. And then this is the patient's bladder. And you can see the bladder is getting rid of the dye that we injected. But all these other areas that I've circled in red that you see are black are areas of tumor. So this patient had very large lymph nodes, cancerous lymph nodes in his neck, as you can see here, in his armpits, and also in his belly. And this scan was taken 3 weeks after this patient had received very high doses of chemotherapy. So this patient's disease was very refractory. 30 days after receiving our CAR NK cells, you can see all the tumors gone. All you can see is the brain. That's normal. These are the patient's kidneys that are getting rid of the dye. And this is the patient's bladder. And what was also very interesting and very important to us was this patient didn't have any toxicities. We didn't observe the toxicities that had been described with CAR T cells. Remember, I said patients can develop high fevers, drop their blood pressure, need ICU care. Well, none of the patients on our trial develop these complications. And despite of the fact that these umbilical cord blood units were completely mismatched with the patient, we didn't have any graft versus host disease. So this was very exciting to us, and that led us to then start asking questions. Well, can we apply this to other cancer types? So again, I'm a hem-oncologist, so a lot of the focus of our research initially was in blood cancers. And so we started a clinical trial in patients with very aggressive type of lymphoma. And these lymphomas, such as Hodgkin lymphoma, and these cancers usually have this molecule called CD30 on their surface. And so again, what we do is we took our banked umbilical cord blood units. We grow them in our lab, and all of this is being done at MD Anderson. We do all of the manufacturing, etc., ourselves. And then the patients were treated with these natural killer cells that had the GPS system to recognize cells, cancer cells that have CD30 on their surface. We treated 42 patients. These patients had had a median of 7 prior lines of treatment, which means that these patients had received chemotherapy after chemotherapy. They'd received antibodies, like these antibody drug conjugates that you just heard of. They'd received checkpoint inhibitors. Many of them had had a bone marrow transplant, and some of them had even received CAR T cells. All of these patients had progressive disease when they came to us. So all of them had disease that was not responding to conventional treatments that were available. And our results were unprecedented in that of the 42 patients that we treated, 93% had evidence of their tumor shrinking. And in 2/3 of the patients, the cancer was completely gone. And that's what we call complete remission. And we didn't see any toxicities. This is a PET scan of a young man with Hodgkin lymphoma. This gentleman had had 8 prior lines of treatment, including intensive chemotherapy, including a bone marrow transplant, and including a CAR T cells. And basically, you can see that he was kind of riddled in cancer, including in his bone. This is his brain, normal. This is his heart pumping the dye, that's normal. And this is his bladder, which is getting rid of the dye. And this is just showing 30 days after the cells, everything is gone. And all you see again, the brain, normal, heart pumping the dye, normal, kidneys, normal, and bladder, normal. So this was, again, very exciting to us. And then led us to start seeing whether we can expand our cell therapies to other cancer types and to target other molecules. And so this is a trial that we very recently started, where our natural killer cells are targeted against another molecule called CD70. And CD70 is present on many different cancer types, including blood cancers and including solid tumors. So now we really have started focusing on solid tumors. So this CD70 is also expressed in kidney cancer. And this is just to show you our manufacturing in a little bit more detail, in that we take the umbilical cord blood from our bank. In the lab, we grow the cells, we engineer them, and we freeze them. And from two manufacturing runs, we manufactured 250 patient doses. We calculated the cost of one of these therapies for us was $600. The patients come, receive 3 days of very gentle chemotherapy, and this can be given as an outpatient. And then we just get a bag of these cells. They are thawed and infused at patient bedside. And so with this particular trial, again, because it was first in human and it was never being done before, FDA were pretty conservative in that they said to us, we want you to start treating patients at a very small dose of just 8 million cells. So just to put this into context, when we do some of our preclinical work in mice, to one mouse, which weighs only 20 grams, we give 10 million cells. So our starting dose level for this particular trial was less than what we give to a mouse. And so I would like to share with you the case of a young man that we treated at dose level 1. So this young man received just 8 million cells. This is a young man with Hodgkin lymphoma again. He had very extensive disease, as you can see here. Again, large, massive tumor in his neck, in his chest, and also in the abdomen, this huge mass in the pelvis. He'd had 7 different prior lines of treatment, including 2 different transplants, one from his mother, so very high-risk type of transplant. And in spite of that, his disease was coming back. And when he came to us, his options were either go to hospice or take part in this first-in-human trial. The day that we infused him the cells, his mom and dad requested for the chaplain to be present in the room with him because we really didn't think that he was going to make it. 3 days after receiving the cells, he was sitting up in bed playing video games. 1 week later, he was discharged home. And this is his scan 30 days later and 60 days later after receiving the cells. And the reason I want to show you both time points, you can already see by day 30, so much of his disease is gone, right? We see the brain, this is his heart, the kidneys, the bladder, but all of these huge masses have gone. We still have a little bit of tumor left here. By day 60, everything is gone. You just see the brain, the heart, the kidneys, and the bladder. And the reason for that is that these cells, as I mentioned, they're living drugs. In the patient's body, they continue growing and they continue trafficking. And so they can persist in the patient's bloodstream for, we've shown, for months on end, you can still detect them. So, of course, we're very excited about this particular trial. The trial is ongoing. We've treated 19 patients, with 15 of patients having responded. The average number of prior lines of treatment for patients on this trial is 8. And for some of our patients, they've had 14 prior lines of treatment. So very aggressive and very heavily pretreated patients. As I mentioned, now we are putting our focus very much on solid tumors. I certainly have a vested interest because of my dear, dear friend. And so we started looking and saying, how can we go after solid tumors? And so we found that there is a marker, a beacon called TROP2, that is expressed on many different cancer types, including ovarian cancer and including pancreatic cancer. But here is listed the cancer types that have this particular molecule on their surface. And in fact, there is already an antibody drug conjugate, sacituzumab, that is being used in the clinic and is FDA approved for breast cancer. And so what we did was we engineered our NK cells now to recognize TROP2. So basically, we gave them the GPS system to go after any cells that express TROP2 on their surface. And this is still early studies. We've done experiments in mice. This is a mouse model of ovarian cancer. And basically, we can follow the growth of the tumor using a strategy that we call bioluminescence. Basically, the tumor cells that we put, these are human ovarian cancer in the belly of the mouse, and as the tumor grows, the color goes from blue to kind of yellow and red. So you can see within 2 months these mice develop a lot of tumor. If you use non-engineered natural killer cells, really, they cannot control the tumor. These tumors are very aggressive. If you give the natural killer cells intravenously to the mice, they don't do a good job of curing the mice. But if you inject these natural killer cells directly in the belly of the mice, so through a port, you can see that we can actually cure a number of the animals. This is again a Kaplan-Meier curve, so a survival curve, where you can see that this one mouse succumbed to the tumor, but the other animals, the other 4 out of 5, were actually cured. But the reason I want to show you is that the route of delivery also really matters. In this particular case, because ovarian cancer and pancreatic cancer are both cancers that live within mostly the abdominal region and the pelvis, we have to develop our introduce our NK cells directly into the region where the tumor is, and there they would do a much better job of controlling the tumor. So now with Dr. Jaziri, and you saw him on the video, he's my good friend and my colleague, we've started a clinical trial. This is for patients with platinum-resistant ovarian cancer, so very advanced disease, where we have generated already these TROP2 CAR NK cells that are frozen, available off the shelf, and then they will be thawed and infused directly through a port in the abdomen to patients. So I'm very excited about this, and I hope I'll have the opportunity to come back and present some of these data to you in the future. Now, however, we do know that the solid tumor microenvironment is very aggressive and and that there are challenges that we need to overcome. And whilst this trial is ongoing, we're also making a lot of additional changes and improvements to our CAR NK cells. So we're looking at strategies to improve the growth, the persistence of the cells, to make them more resistant to the microenvironment of the tumor, because ovarian cancer, just like pancreatic cancer or brain tumors, it's the environment is very hostile. The cancer cells try to live, and they compete for nutrients, they compete for oxygen with your immune cells. So we are trying to make our natural killer cells fitter. We are looking at ways to make our the tumor cells more susceptible to NK cell killing, and also looking at how we can even engineer our natural killer cells with multiple CARs, multiple GPS systems to recognize different tumor types. So this is basically just summarizing all the clinical trials that are currently ongoing at MD Anderson for different tumor types. We have 19 different trials that are up and running now for different cancer types, including blood cancers. I mentioned pancreatic and ovarian cancer, colorectal cancer, glioblastoma, which is a deadly brain tumor, uveal melanoma, and also more recently, we've extended our approach for the treatment of autoimmune diseases. And this is an incredible collaboration across MD Anderson. We work very closely with the clinicians who have expertise in the diseases that they treat. As I mentioned, I'm a hem-oncologist, but I work very closely with Dr. Jaziri for ovarian cancer. And I think this kind of collaboration is very, very important as we develop these new drug therapies for our patients with different tumor types. And I just want to say, the future of these therapies is very bright. We live in an incredible time in science and medicine. There is so much data that is coming through, so many breakthroughs, so much in terms of understanding of tumors, tumor susceptibility, tumor resistance, why, how, single cell data onto why it is that some tumor cells are being killed by our approaches, some are resistant. The promise of artificial intelligence and machine learning that will really help us expedite the development of novel therapeutics for our patients. So with that, I just want to very quickly acknowledge my incredible team. of young scientists who are working day and night with just the focus of developing therapies for our patients with cancer, our cell engineering group that are manufacturing the products, my incredible collaborators across the institution, all of our funding agencies, including NIH and the various philanthropy groups, the Marcus Foundation that's actually supporting our ovarian and pancreatic cancer trials, and most importantly, our patients and their families for their courage and for putting their trust in us and participating in our first-in-human trials. And I'm very grateful for your attention and looking forward to the panel discussion. Thank you very much.